While you join, I'd like to welcome you to the Gabriella Miller Kids First Pediatric Program Spring Webinar. Today, you will learn how big data advances pediatric research. I am Marcia Fournier, the program manager for the Kids First program. I came from Brazil to the US to pursue my education in cancer biology about 27 years ago. Since then, my experience with big data and clinical diagnostics brought me to the Kids First program a little more than 2 years ago. I will be your master of ceremony today. So I will start with a few reminders and housekeeping items. This event will be recorded. So for those who may be uncomfortable with being recorded, please note that a recording of the event will be made available on the Kids First website after its conclusion. Please remain muted through the event. Send your questions ahead for a dynamic discussion if you'd like. If you have questions, submit them through the chat box. We would like to ask you to respond to an event survey, which will help us to shape future events. Check the link in the chat box. A little bit about the Kids First program. Before I start, let's see. Okay, many of you already joined. Thank you for joining. The Kids First is a common fund program. The program aims to accelerate research with data sharing across pediatric conditions, including structural birth defects and childhood cancers. 10 years old Gabriella Miller, a childhood brain cancer patient and advocate, inspired the program authorized by Congress in 2014. Since 2015, the program has granted $12.6 million per year to develop a data resource for pediatric research. Birth defects and cancer are top childhood death causes. Studying these conditions together makes sense, though common genes and pathways and higher cancer risk in children with birth defects. Kids First resources allow sharing multiple data types across these conditions, which enables a better view of the condition underlying biology. The goal is to speed up the development of diagnostics, treatments, and prevention tools. Data achievements include 33 studies released with over 27,000 genomes of patients and families. Nearly 700 data access requests has been approved for secondary use of the data to date. Kids First is also collaborating and developing ways to cooperate with other data science resources at NIH and outside. Now, let's go for the agenda highlights. We will start with a panel on big data in pediatric research and follow with a demonstration with a deep dive on how to use Kids First data. Our panel discussion goal today is to provide to you an example of primary and secondary use of the Kids First data. We will learn how our researchers are using the data and how this data is leading to discoveries. We aim to inspire you to explore the Kids First data and use it. So I will start by calling our moderator, Adam Resnick, Director of Center for Data-Driven Discovery in Biomedicine at Children's Hospital of Philadelphia. His research is focused in pediatric brain tumors. Hi, Adam.
Hi, Marcia. It's great to be here.
And our panelists are Dr. David Teachey, oncologist at Children's Hospital of Philadelphia. His research focused on investigating novel therapeutics for pediatric T-cell acute lymphoblastic leukemia, among other pediatric conditions. His goal for today is to understand how big data and large-scale genomic profile efforts, such as those through Kids First, help us not only to better understand why some patients develop cancer, but also help us translate new treatments to the clinic. Thank you, David, for being here today with us. Dr. Soumya Rao. I would like to call you to the podium. She's a postdoctoral researcher at the University of Missouri, Kansas City. Her research focuses on non-syndromic orofacial clefts using genomic data. She aims to inspire researchers to fully utilize Kids First resources, to advance discoveries in pediatric genetics, and to offer parents and children hope for future diagnoses and cures. I'm looking so much forward to hear your discussion. And Adam, the panel is all yours. I will pass the baton to you.
Thanks so much. And, I think I'll have a very easy job being a moderator with our guest today. We'll start the panel off with really two brief presentations by the panelists, and then I'll guide, moderate discussion with some questions and hopefully thoughtful answers in response, as well as please submit questions via chat throughout the discussion or presentation. So we'll start with Dr. Teachey first with the presentation, and we'll pass the baton to Dr. Rao.
Thanks, Adam. And I'd like to really thank the Kids First organizers for having me here today to talk for a few minutes on the work that they've supported. Huge fan, Kids First, huge fan of the NIH and how much they've really helped this research and other research. I'm going to give a very quick overview, 2 slides. My lab and my clinical research are focused on acute lymphoblastic leukemia, which is the most common cancer in children. I think that the improvement in cure rates for kids with ALL is one of the greatest success stories in modern medicine. 70 years ago, everyone with this disease died, and now we're curing over 80% of children and young adults with acute lymphoblastic leukemia. Most of those inroads really happened, though, through optimizing old cytotoxic chemotherapeutics. So we're still not curing everyone, and our therapies are pretty toxic. ALL has 2 types, B and T ALL. That was mentioned before. My main focus is T ALL. And over the past 15 years, there's been a lot of major strides in the understanding of B ALL. And a lot of that's been through genomic profiling. It's allowed us to identify targeted therapies, magic bullets, as you were, like tyrosine kinase inhibitors, imatinib, which are now helping patients in the clinic, and also immune therapies. So immune system's a part of our body that fights infection. And immunotherapies are things that are trying to act like the immune system to attack cancer cells. And through this, we have lots of new drugs for B-ALL. In TLL, we are way behind that and not have made the same advances, but I think based on the data we now have for this large Kids First project, we're able to start doing that as well. So this is a large collaborative project between the Children's Oncology Group and St. Jude Children's Research Hospital, where we're trying to move the needle in TLL. It's led by myself and Charles Mulligan primarily, but also a lot of other physicians and scientists. The red people on there are all the names of the people who are involved in this project, including many very talented trainees and postdocs, including Petri Polonen, who did a lot of the genomic stuff I'll show on the next slide. And we're also tackling this through Kids First and through other NIH funding in many ways. We're looking at it at the DNA level, the RNA level, the protein level, and we're integrating with other work. There's dozens of physicians and scientists who are working on this. The Kids First Award sequenced 1,309 children of TLL treated on a single trial, the ALL-0434 trial, which is an NCI-sponsored trial through COG. And this included sequencing of tumor and non-tumor normal cells. Next slide. And this is just showing one snapshot of some of the data we have, which were recently accepted into Nature and should be coming out this summer. On the left is a UMAP, which is looking at the transcriptional profiling of each dot as a case. We're able to classify TLL into 15 different groups now, so we finally can actually start splitting TLL so we understand the biology better and use that to identify therapy. And on the right is showing how for the first time ever, we're able to integrate clinical data with genomic data and response data so we can actually predict which children with TLL are more likely to be cured and which children of TLL are less likely to be cured of our current therapy, allowing us to reduce therapy for some of the very favorable groups or try alternative therapies in some of the high-risk groups. And with that, I'll stop and turn it over to the next speaker.
Thanks so much, David. That's very exciting, and congratulations on the recent acceptance. Let's turn over to Dr. Rao.
Hello. Hi, Adam. Hi, Marcia, David, and all. I hope my screen is visible.
It is. It's not yet in presenter mode if you're able to present it.
Yeah. All right. So
then- You might want to just switch the display setting on the top so that we're currently seeing the presenter mode on our side.
okay,
It's
let me see.
under the display setting, top left. You can just click on duplicate. Perfect.
All right, thank you. Thank you, Marcia, for inviting me here. I'm very excited to share how I used the valuable resources from GMKF for my own research. And I hope I can inspire others to use it too and provide some hope to the parents for rapid diagnosis and novel cures or prevention. So I'm a postdoc in the Cox lab at University of Missouri, Kansas City. And our lab mainly focuses on craniofacial birth defects, mainly cleft lip and palate, as well as craniofacial microsomia. We have large multi-generational families with multiple affected members who display a spectrum of phenotype in both these conditions. We can see very mild to very severe cases within the same family. And in case of clefts, it could be unilateral, bilateral, a partial cleft or complete cleft, including palate or without palate. In case of craniofacial microsomia, we have seen cases with very mild phenotype like a skin tag, ear tag, and small ears, low set ears, and also anosia, like there are the ears are external ears are completely closed. And in severe cases, we see uh, eye epidermal bulboids and uh, mandibular hypoplasia, which gives the facial asymmetry. For both these conditions, we have mice models as well, which mimic the human condition so well. So I use Gabriella Miller Kids First cohorts of orofacial clefts, where there are more than 1,200 trios. And these have whole genome sequencing data available for us. And they include 4 different populations. And it is much bigger than our cohort. For craniofacial microsomia or microtia, we have 255 trios. And most of the times we use it for finding genes, novel genes, with the rare potentially pathogenic variants. And I also use to validate the variants in the novel genes of interest, a list of candidate genes, which I found through in our cohort, and use GMKF cohorts to validate them, to find additional cases, additional variants in the same genes, and it helps us prioritize them for experimental validation. If we find loss of function variants, it is pretty straightforward. We immediately can look up and see what pathways and what functions these genes have and how it could be affecting the craniofacial development and prioritize them for experimental validation. But in case of missense variants, we need more cases, and those can be, those can give us a prompt to take it further and do some experimental validation, the mouse model or zebrafish studies to show that they have some effect in craniofacial development. So one such study we published last year where we found a mutational hotspot in one new gene, immortal1, and also defined a new syndrome with orofacial clefts, cardiac anomalies, and tall stature. There were other phenotypes as well associated with some of the patients. And here we can see a hotspot in this region at amino acid 157 to 161. And it is a very conserved region across the species. But it took 20 years to come to this stage when we had the first patient that was seen 20 years back. And after a few years, the next genome sequencing became available, and clinical sequencing helped us find this one patient. But we didn't have any additional support. Over the years, more and more collaborators and people through Gene Matcher, they gave us additional patients and we had a larger cohort. And but now I think if we had GMKF, then we would have found 5 additional patients. And it is out of 876 isolated non-syndromic cases from 3 different cohorts. At that time, I had only access to 3 different cohorts, and the Philippines cohort was added later. But I could filter my variants based on population frequency and have some conservation scores as well. So it would have, we eventually did the experimental validation, and we showed that this gene is affecting the YAP signaling, and that's how it is contributing. But I wish, now I know we can accelerate discoveries. This is the Kids First portal I use thoroughly. And as I told you, the phenotype has, it is a new syndrome and it has cardiac anomalies as well. So I could see multiple new variants, and the studies we have in the corner, most of them include congenital heart diseases. So it gives us more confidence to go ahead and put that as the gene associated syndrome. This is another gene. It is a very ARH-GAP35. It is a well-known cancer gene, but it was also reported to be involved in developmental delay and intellectual disability, to cause some ocular phenotype and eyes. And we found in our cohort 3 different cases with the variants in this gene, and additional 8 cases from GMKF cohort. So this is the next gene that we are going to publish, and we have a whole list of novel genes and maybe novel syndromes which are associated with multiple organs but have the common pathways and common function. So I hope I could show you how immensely helpful Kids First resources are for me. With that, I thank you. And this is our Cox lab. We are in Kansas City, which is famous for Kansas City Chiefs. We are on the map because of them, and also because of the Cox lab. We have our collaborations with the Children's Mercy and Star Wars, where we send our samples for sequencing and also validate our results on zebrafish models. And we are thankful for them for collaborations. Thank you again, Adam.
Thank you. And, you did a really great job, Dr. Rao. And I think you actually set the stage perfectly for the next half of the session, as you'll see. Both of you actually started sort of at the opposite extremes by highlighting essentially size as, a challenge. David, you started really, highlighting that you had assembled one of the largest cohorts of samples that required characterization analysis in order to drive new knowledge. And Dr. Rao, you highlighted that although you had existing cohorts, that they were too small and insufficient to really drive translation. Let me start with you, David, briefly, because the context here is so different. Maybe you can just briefly describe how it is that such a large cohort comes to be, for those who are not necessarily, fully aware of the COG, and maybe highlight in a certain way what would have happened if there wasn't, Kids First as a resource, and then I'll have a follow-up question to that.
So the samples for this were banked for the Children's Oncology Group. So the Children's Oncology Group is a cooperative group, which is a NCI-sponsored group whose mission is to try to cure more children with cancer. It was formed by a bunch of other smaller groups merging around the year 2000. And it's currently across North America, Australia, and New Zealand at over 200 different hospitals. Childhood cancer is rare. Very rare. There's not that many kids, fortunately, who get cancer every year, and you're never going to be able to cure it by just having each center do their own thing, which can sometimes happen with adult cancers that are more common. So decades ago, people pooled things together to try to treat it the same way, perform clinical trials. And as part of that, COG and the NIH really invested in biology so we can understand more about cancer. So it's not just a trial to test one new drug. And the trial in question that Kids First funded the sequencing on was asking a new drug question, but which improved cure rates. But beyond that, every single patient had the option of having samples banked, and of 1,500 patients on the trial, 1,300 of them actually had samples banked over almost a decade, making it the largest cohort of patients with this particular type of cancer to have samples banked. And it really helped us unlock a lot of understanding on the biology of T-cell acute lymphoblastic leukemia that's allowed us to make some inroads. I mentioned before, ALL has two forms. It has a B form and a T form. What we found years ago for B-ALL is most of the genetic alterations that can actually cause cancer are in coding parts of the genome. So for the non-scientists out there, our DNA is very huge, has billions of what we call base pairs. Less than 2% of it is actually what we call coding that actually makes protein. The rest of it's what we call non-coding, which we used to call junk DNA, but now we know that's what actually turns different genes on. So it says, should you turn on this gene if you're a heart cell versus turn on this gene if you're a hair cell? And that's very important for cancer, depending on the type of cancer. So in B-ALL, most of the alterations were not in the coding, were in the coding portion. In T-ALL, what we found was most of the driver alterations that were causing cancer were in non-coding alterations, over 60% of the cases, and it was highly complex. It's not one gene that's changed to cause cancer. It was hundreds of different genes in hundreds of different ways. And one of the reasons we've really never been able to make big insights into TLL biology and understand what genetic things correlate with outcomes so we can develop better therapies is we've never had the scale to look at it at such a high level. And it needed that scale to understand the complexity of the disorder, and also needed the whole genome level, which Kids First allowed to happen, to actually bind over 60% of the different genetic changes that were causing T-cell leukemia. So without Kids First, this work would never have happened, because doing whole genome sequencing on 1,300 patients, their tumor and their germline, is not cheap.
Thanks so much, David. And I think you really highlighted exactly what I'd hoped you'd point out, which is that, despite the fact that, a dominant framework of engaging cancer really focuses on coding mutations and panel-based diagnostics, really the transformative power that you've been able to harness is underpinned by whole genome sequencing. I'll ask you one more question, and then I'll go to Dr. Rao. You're also fairly unique, within the context of Kids First and more broadly, in that, it really looks like you've layered multiple layers of data modalities together, right? Whole genome sequencing, RNA-seq, methylation, proteomics, phosphoreporting. It seems like essentially, the first phase of that work, from what you presented, has allowed you to risk stratify patients in ways that will likely change the care that they receive. What do you hope is the next set of activities and layers, associated especially with, the proteomics, phosphoproteomics, and other data will bring to bear?
So first, I would say that we need to validate some of our findings at the genomic level. And fortunately, Kids First funded a second award, which is actually sequencing the successor trial, so we can actually see what's the same and what's different. They also had supplemental funding to look at the protein level, because what really matters is, are these changes we're seeing in the DNA actually impacting RNA and then actually translating into protein and functional protein? Because we can make guesses, but we really can't necessarily establish that. And also looking at the epigenome level, trying to say, even though we're predicting this gene may be turned on, it may have epigenetic changes like different methylation that's actually keeping it off. And one of the reasons we could do all this work, so the genomic work, the proteomic work, the epigenome work, it's all been funded through the NIH for one mechanism or another, and it gets to be almost self-fulfilling in that I have taken the first Kids First award I had and turned that into 2 R01s with collaborators, basically because the NIH finds this whole work, new discoveries. Now we need to actually tackle some of these discoveries, and this is how we're going to do that. We're going to look at mechanism. We're going to develop novel treatments. So it launched the ability to do all this other work that never would have happened in the first place if we weren't able to do the initial sequencing. So it's very exciting. And again, I can't thank the Kids First group enough.
That's awesome. And again, I think you're really an exemplar of, the mission for Kids First, which is really to translate, these data into clinical impact, right? And, the fact that you can point, to a publication that changes the course of care for patients as a result of this work is, amazingly significant. Dr. Rao, I think you're, probably at the, not only, began with smaller amounts of data, but you're also at a different point in your career than someone like Dr. Teachey. I love that you pulled up, the slide showing the Kids First portal. Maybe talk a little bit about what it means to a young investigator to have access to data, and what would it look like otherwise, for you as a young investigator from a career perspective a bit, on behalf of this type of effort.
Yeah, as you said, as a young investigator and from a small lab, UMKC doesn't have high performance computational clusters or servers to store all this data. So whatever data we have generated through our cohorts, it has been located at different other clusters. And we have collaborations in Australia and others. So I extract only the data that I need and I use. But here with Kids First, we have Cavatica, a cloud platform, where and also access to, as I said, huge resources, whole genome sequencing. And most of the times, I use Cavatica. It is a cloud-based platform, so I can access this data from anywhere, from home, and just I need to have a login and password. And I also I'm grateful that there is access to other databases as well. In Kids First portal, I can see and cross-reference all other cancer and congenital birth defect databases. So which gives me, the analysis part becomes like the major focus instead of discovery part. So it has actually, like when I joined in 2020, that is when I got introduced to Kids First. I think I attended the first webinar as well. And since then, I've been engaged with, through monthly office hours. And I have also been involved in beta testing, thanks to you. So whatever technical glitches were there, whatever loopholes were there as a user, we could fix them, and what information we wanted, that could be added. And it has the recent update. I'm sure I'm looking forward to the demo. But when I checked, everything that I mentioned that I said was lacking, or it was fixed, and it is now very user friendly, and it is very accessible. We don't need to be bioinformaticians to access this data. We need approval. Like for my cohorts, we need approval from the submitters through dbGaP. And but then other databases we can access, which are available for public.
And you'll see from the presentation from the Kids First team in a second that, really we took the publication you highlighted to heart, because you actually inadvertently or purposefully made some recommendations as part of your publication in ways that hopefully you'll see fulfilled. Let me now end over the last couple of minutes on, one of the key parts that, Kids First tries to really foundationally establish and is, truly unique, which is that we are essentially supporting investigators spanning congenital abnormalities as well as cancer. And let me start with you, David, briefly. If you can just comment on maybe two aspects. Most cancer researchers still, in many ways, think about cancer as a somatic disease. I wonder if maybe you can comment on, that framing in the pediatric setting, in part because, very few congenital defect researchers think of somatic components. And then secondarily, if you maybe can comment on the intersection between leukemia and other syndromes more broadly, perhaps Down syndrome or other contexts like this? Again, highlighting some more from the COG recently.
So, great question. Tough question. I mean, I think it shows how much one thing is it shows how much the field has evolved over the past 20 years. When I started doing pediatric oncology on the clinical side, we would go into families' rooms and say, childhood cancer has no genetic component whatsoever. It's bad luck, your DNA changed, and you develop leukemia. We don't understand why. And now we're learning more and more that there are some predisposition in at least 10% of patients to have cancer as their kids. Kids aren't supposed to get cancer. I mean, if you take a step back and actually think about it, there has to be a reason it's happening, and it's probably a multi-hit phenomenon where you're born of something that may put you at risk, and then it's the right exposure or the wrong exposure to a virus or something else that triggers your immune system, and it leads to a perfect storm, and it leads to childhood leukemia. So I think to understand cancer better, we need to figure out what drives it, and then maybe we can figure out ways to prevent it. So if we understand what things in our germline DNA or make us who as we are may predispose to cancer, we could either screen for it or even think about ways of using gene editing or something 10 years down the road or so to actually prevent it from happening. I mean, as a cancer researcher who studies leukemia, where a lot of scientists and physicians are all siloed. I work on my disease and this model, this person works in that disease and their model, and there's a lot of overlap in science and medicine over things that we learn in one disease really apply to others. And through these large data sets, it lets us integrate all those data so we can try to understand not just the disease of interest for that particular person, but more how disease evolves generally. And childhood cancer in particular, which is so rare, we really need that multimodal across disease, both looking at the tumor itself and germline ways they can interact so we can understand the disease better. There are definitely, going back, I mean, some people here in the clinic noticed decades ago that some patients who develop cancer actually probably have some syndromes or birth defects that correlate or associate with it. As well. One person who was the chief here, Anna Meadows, for a while, published a paper in the New England Journal of Medicine where she just noticed that some patients with Wilms tumor had shorter legs, which was an early insight onto hemihypertrophy, which led to understanding the biologic manifestations of what's the genetic change that lead to both the tumor and the congenital anomaly as well.
Perfect. Thanks so much. Dr. Rao, I'll invert the question to you a bit. So you, in one of your slides, you showed essentially the mutational profile of a gene. And to many cancer researchers, the profile that you showed looks very familiar because it almost looks like a gain of function context where there's a selective pressure for a particular region to be mutated in that setting. Most of the time when we think of congenital defects, we think of loss of function sort of context. Maybe you can talk a little bit about, the challenges and opportunities of thinking about gain of function versus loss of function and how you're thinking about it in the context, of craniofacial research. And perhaps those are mechanisms by which, again, there might be some synergy between approaches that cancer research takes and craniofacial research might take. And correct me if I'm wrong on my assumption.
Definitely there is, I cannot comment on gain of function yet, but we could see most of the variants that were concentrated in one region, they were causing cleft and congenital heart defects and tall stature, and others which were now we have seen more patients like from GMKF cohort only. They are everywhere in the gene, in the coding region. So the phenotype which is associated, the secondary phenotype could be variable, right? But as you said, we are mostly looking specifically at the loss of function, truncating mutations, truncating variants. So most of the times we don't find such straightforward, truncating variants just like that, and we give a diagnosis. So that's why we have only 10% molecular diagnosis for congenital birth defects, especially like in our cases. So my aim is to look at the entire genome and not rule out anything. Like uh, I was told by an expert once, like all the genes that are to be discovered, they have already been discovered in because cleft is such a old field, and I still keep discovering novel genes. And the subtle mutations, like non-synonymous changes, they change the protein structure somehow. They've changed the interaction with other proteins, and most of the times we can't report them just like that unless we have a large number of cases, and we do some experimental validation or just a prediction like in silico predictions. So here, like I would, most of the cases we have are coming from the clinical sequencing, where they do only exome sequencing, which is only looking at the coding region. And with this whole genome sequencing data, I have access to the whole non-coding region, which is like 98% of our genome. And there are changes which can affect the regulatory region, maybe splicing, maybe some access to the transcription factors and others. So, and there could be multiple compound variants in cis or in trans. And in one of the cases, in one of the genes around that region, we found a haplotype. It is in submission. It is a gene. I don't know if I can take the name yet or not,
Don't
but
worry. Yeah.
yeah, but we found a haplotype. If it was associated with the craniofacial microsomia only in the probands, in parents and in control population, that haplotype was not associated even with the lead variant. So
That's
that can
awesome.
only be,
Yeah.
yeah, that can only be discovered through multiple cases and whole genome data.
Well, thanks so much, for sharing that next phase of your research. And, you both did an amazing job highlighting your work. And importantly, really, the power of both sharing data and what's to come. So thank you both for all your comments. I had a list of probably 7 more questions, but we ran out of time on this part of the presentation. But please hang around and to see what is really an exciting next phase for the Kids First program in ways that I think sets a stage for some of the questions and points you guys made on the opportunities coming next. All yours, Marcia.
Thank you, Dave, Soumya, and Adam for this amazing conversation. It's always great when we leave with a taste that we want more. And so I think that is the goal here. I really want to highlight some of the points that you drove into how Kids First is bringing a diverse data that can advance therapeutics and discoveries across pediatric conditions. I think also, thank you for highlighting some of the things that we talk about, how studying cross conditions can empower your research by finding genes that maybe were important for other condition, and now you find that is important for your condition, and then you can cross-pollinate this knowledge and advance new discoveries. And also, so with that, we'll have to move on from the panel to our next section. So thank you again for your contributions in the discussion and giving your perspectives, Soumya as a secondary user and Dave as your primary user of the Kids First data. And for the audience, if you got inspired, stay with us for the second part of the webinar, where we're going to have a practical demo on how to use the data. But before I go and introduce our next speakers, I want to highlight NIH funding opportunity that will empower the analysis, primary analysis and secondary analysis of Kids First data. So the first opportunity is called the Small Grants for Analysis of Gabriella Miller Kids First Pediatric Research, is a RO3, and we will have the link to the PAR at the chat box, so you can check it out. This opportunity aims to engage the research community in enhancing Kids First data value. The goal is to increase data value through development of new tools, analysis, and data enhancement. So these opportunities support small projects, analyzing Kids First genomic and phenotypic data, allows 100,000 per year for 2 years, and no preliminary data is required. The potential areas of your proposal could be primary or secondary analysis of Kids First data, development of a new analysis tools and workflows, refinement of diagnostic techniques, discovery of treatments, new target treatments, and additional longitudinal data. So these are topics that are relevant for this opportunity. And the next application receipt date is June 16, 2024. But also check for future receipt dates. In 2024, we have additional date in October and 3 dates in 2025 for the same opportunities. So check on the chat box. And then the second opportunity is also our 3, sponsored by the Common Fund. The funding opportunity has the goal to enhance the utility of Common Fund data sets, and Kids First, part of the Common Fund data ecosystem, is relevant for this opportunity. Check in the chat box the RFA, and the application date is June 27, 24. Okay, so with that, if you have questions either to the panel or questions about funding opportunities or Kids First program, feel free to add to the chat box and we will be answering in written or orally. I will now move to give a quick introduction to the next speakers. Our demo has the goal to give you a deep dive on the Kids First data, specifically on the variant browser to empower your research. So Dr. David Higgins, informatics program manager of the Children's Hospital of Philadelphia. He works with the community to accelerate the transition from data to knowledge to impact. Welcome to the podium. And Dr. Jared Rozowsky, technical program manager at Velsera. He works closely with the Kids First Data Resource Center to design, implement, and utilize new features on the Cavatica platform. So Dr. Jared hopes to get people excited about the possibilities of using Kids First data and technology to uncover new insights that may change someone's life. So with that, I'll leave you with our next speakers, and I hope you enjoy the demo. David?
Thanks so much. Thanks so much, Marcia. Great to be here. So I'm so excited today to present here at our Kids First Spring webinar, showing about variant discovery and Kids First data sets. So I'm going to do the first half here, and then I'm going to pass over to my partner, Jared, to share a little bit of an additional deeper dive as well. But before we do that, I do want to acknowledge that Jared and I get to stand up here and present this exciting new updates and new resources, but behind us is a team of dozens of people who work very, very hard to make all of this possible. So just want to acknowledge all of us at the Kids First Data Resource Center. Everybody has put a lot of effort into this presentation, what we're about to share today, and I wanted to acknowledge them and thank them for all their hard work that's gone into this. All right. So if you haven't been to explore the Kids First website recently, the first thing I wanted to share is that we have not just a new portal, but also a new website. And so if you navigate to kidsfirstdrc.org, you'll see a completely updated page with new content. And it really does a great job of highlighting what we are, what we do, and the value of what we've built. So after the webinar today, if you haven't seen it, please check out our website, kidsfirstdrc.org, and read more about us. Of course, and additionally supported with that is that we have a whole new help center. And so attached to the website is guides and with pictures and screenshots and short little videos about how to use our portals and platforms if you ever get stuck and need some further help. So today we're announcing that we're launched a new version of the Kids First portal. And so the portal is available at portal.kidsfirstdrc.org. highlighting that we have our previous version of our portal will soon be going away here coming up in the middle of June. We're going to be depreciating that older portal, and we'll only have the new portal. So some of the new features you'll notice right on the home page is that we now have an updated kind of underneath the layer of the portals that we have updated servers. So now the Kids First data is stored in what's called a Fire server, which allows for API authorization for advanced users. We've also enabled connections with the Cavatica variant workbench, which we're going to hear a little bit more about from Jared in the second half of the demo today. Up at the top, we've improved the studies page. So now the studies page includes additional detailed information about the different data types and experimental strategies of the different data within each study. And the data exploration tool has been improved because we've combined the data exploration and file repo pages into one single page. And so now you can search for participant file specimens and data files all on one page, which I'll show here in my demo in a couple minutes. We've also made improvements to our variants page as well. And so the variants page allows you to be able to search and explore variants in the Kids First data sets and gives you summary level variant exploration. So we'll show a little bit of an example of how to use the variant search tool in our demonstration. And so there's going to be two parts today. So my part is part one, where we're going to be exploring the data on the Kids First portal. So we're going to try to identify participants, look at genomic variants, and then figure out how to review studies and apply for access. And then after that, I'm going to pass some data to our cloud platform, Cavatica, and I'm also going to pass the baton to Jared, and he's going to show us how to use Cavatica's variant workbench to identify genomic variants within those participants, and then pair the variants with annotations as well. So a demo, I think, is even better when we have a specific example, and we're so fortunate that Dr. Rao gave that great background about orofacial clefts. So I don't need to go into too much detail because I am a little short on time, but orofacial clefts are caused by the failure of tissue to fuse properly during fetal development. They're a relatively common birth defect, can cause as a result of both genetic and environmental causes, and multiple developmental pathways have been implicated in orofacial clefts. And so as Dr. Rao shared in her intro, she recently was part of a group that published a paper identifying the gene AMOTL1 and variants in it associated with not just orofacial clefting, but also cardiac anomalies and tall stature. So her and her research partners used the orofacial cleft cohort data sets as a secondary cohort to kind of validate their findings. And so again, to highlight the example here, so they were able to identify this hotspot here in positions amino acid 157 through 161. And so at this time, and so to pull out a specific quote from the article, Dr. Rao and the authors identified that identifying the individuals that have these variants were difficult at the time. And so as a result, they needed to download the trios in order to be able to find participants that had that variant. So what we're going to show today is that you can now do that on the Cavatica platform and the Kids First portal to get started. So we've improved the process for users in the future. And so the demo that I'm going to share today, if we have a more generic kind of view of it, is a lot of the questions that we get are what types of participants in Kids First data sets are diagnosed with my condition of interest? Do participants in Kids First have variants in my gene of interest? And how do I apply for access to the genomic data for these participants? And so we're going to answer that by showing 3 different pages. So we're going to answer the first one by looking at the newly designed data exploration page. We're going to look at variants in the genes of interest using the improved variant exploration tool. And then we're going to explore the genomic data and how to apply for those by looking at the updated studies page. And then again, we're going to use our example. So today our condition of interest that we're going to look into is orofacial clefts, and our gene of interest is going to be AMOTL1. So at this point, I'm going to navigate to our website. And so anyone can do this following the presentation today. Navigate to portal.kidsfirstdrc.org. And when you navigate to the dashboard, so you notice that everything, it has a different look. We have a new sleeker dashboard with some similar kind of cards or widgets that explain different aspects as far as your authorized data and your connected Cabatica projects. But I'm going to start with the data exploration. And so our first question was, we want to identify which participants in Kids First are diagnosed with my condition of interest. And so we can do that. If we scroll down, originally it gives you a big overview of different data modalities and data types and information about phenotypes. But we can search specifically for, if we go into our clinical tab and apply a clinical filter for our HPO term for oral clefts. So we use on our portal, we use the HPO, Human Phenotype Ontology. And so if I type in oral cleft, then it pulls up oral cleft, which is kind of a parent term that includes both cleft lip and cleft palate underneath it. So then I can apply that term. And so it tells me that we were able to identify 1,517 participants who are labeled with that condition. If we scroll over to the participants tab, so now without actually changing the page, we can just switch between these different tabs. We can learn more about these different individuals. And so most of the individuals with the orofacial cleft phenotypes are in our Kids First orofacial cleft studies. So like the Latin American Family Study or the European Ancestry Study. But you'll also notice that there are additional studies in here as well. So for example, this is the Kids First intersection of childhood cancer and birth defects. So supporting that, the Kids First data set is well annotated across multiple studies for our different phenotype conditions. And so you can use these additional filters to explore what different studies are available or what studies have participants with these particular conditions. So if we look at our study name, again, the most frequent or largest studies with the largest number of participants, again, are the orofacial cleft studies. But we do have participants in the smaller number of participants in our other studies, like our chromosome 18 copy number study or CHARGE syndrome study. So they're to kind of connect the conditions across the multiple different Kids First studies. From there, you can then click across to learn more about the biospecimens associated with those participants, or into our data files where you can then access the individual alignments and variants that are associated with these participants. So I'm going to skip ahead to our variants tab here. And so this lets you search for particular variants of interest. And so, for example, if we wanted to search for a gene, for example, we could type in the name of our favorite gene. And so we might try to look into that study that Dr. Rao shared, and maybe we're interested in AMOTL1. And so we can apply that filter, and then it pulls up a list of variants that are present in Kids First data sets in the AMOTL1 study. At this point, and it's sorted by deleterious consequences. And so many of these are, the most deleterious at the top, and then the missense ones, and then silent ones at the bottom. But we can filter this further. So we can add, for example, an additional filter looking at pathogenicity, looking at CAD score. So CAD score is a filter that predicts how deleterious a particular mutation is. And so if we apply a filter for where your CAD score is greater than 15, and then we want to combine those. So we're looking for variants in AMOTL1 where the CAD score is greater than 15. We get a total of 125 variants. At this point, we can kind of scroll through, and so they're sorted by position in the genome. And so you can see we can actually, if we keep going further and further, we can find that variants in that particular region at the 157th amino acid. So we were able to find the specific variant that Dr. Rao's paper identified, and we're able to learn that there are 2 participants in one Kids First study that have this variant. So we can click on this link and learn a little more about this particular variant. It tells us more information, more annotation information. It tells us about the frequency and explains that in the Kids First European Ancestry Study, there are 2 participants with this particular variant at this site. So if we want to learn more about those participants, though, which 2 participants are they? This is kind of the extent of where we can go on the Kids First portal, because at this point you would need to apply for access to be able to identify who those participants are. So you could do that by going to the studies page, which has a list of all of our Kids First studies. And so if you search for that code, that orofacial cleft of European ancestry, then it pulls up our study here along with the dbGaP ID. So of the 1,400 participants, only 2 of them have this particular variant. And then you could apply for that access by clicking this link, which takes you out to dbGaP. And then you can submit an application to request access for this particular data set. So once you have access, you'll be able to access all of the genome files for this study in Data Exploration tab. But you can also will be able to explore them within the Cavatica Variant Workbench. And so this will allow you to be able to search very rapidly and add these annotations quickly to be able to understand, identify who those participants are. So at this point, you would be able to click this Launch in Cavatica button, which will take you into Cavatica. And so as we launch it, I'm going to pass the microphone now over to Jared, who's going to explain a little bit more about what the Variant Workbench is, how it works, and we'll see a quick demo of identifying variants in the workbench.
Awesome. Thank you so much, David. I can take over sharing screen if that's okay.
Sure thing.
Cool. Slideshow. I feel very fortunate to have such a great panel of speakers today. I also feel fortunate to go at the end because it means everyone has such a rich background in what we're talking about, both in terms of the scientific use case and just what we're trying to convey with the portal and new features. So what I'm going to be talking about for the last few minutes is the new feature that we're calling the Cavatica Kids First Variant Workbench. And the Variant Workbench that we're talking about and that I'll demo was designed specifically to address the limitation highlighted in Dr. Strong and Dr. Rao's paper, where they said the identity of the individuals harboring each variant cannot be determined in Cavatica at this time. We recognize that this type of analysis was a challenge, and we've been working on this feature for the past year, so I'm very excited to be able to show it off today. Generally, the Variant Workbench supports querying across hundreds of millions of variants. You can actually see on the portal that we currently have over 321 million variants within the Kids First studies, coming from over 17,000 participants with whole genome sequencing. The Variant Workbench allows you to combine features such as variants and phenotypes across cohorts, providing the patient-level data, so that controlled access data that David was just talking about. In other words, what we want to know is which participants have a variant or a set of variants, and what are the characteristics of those participants. And we can do all of that in Cavatica because we have RAS enabled and can check dbGaP access to control data at the time that access is requested. Doing a shallow tech dive, I don't want to get too deep into the kind of what goes behind it, because I want to talk about what this enables rather than how it's built. The Variant Workbench is an extremely powerful tool. It has a familiar Jupyter Notebook front end with a Spark cluster running in the back end. The files are stored as Parquet files, which are partition files and are optimized for storage and compute in a Spark environment. Using Delta tables, we can manage versioning to help with reproducibility, since we know that this data is going to change over time, but researchers may want to go back to recreate a paper that they read, and all of this data and change logs will be kept there. And I keep calling them tables because I think it's helpful to think about data in this context. Within the Variant Workbench, when you want to add an annotation, conceptually, you're just adding a column. Or when you're filtering, conceptually, you're just subsetting the rows of that big table. And by framing it that way, at least for me, it makes thinking about the analysis a bit more simple. I alluded to this on the last slide, but there are really 2 components that make up the Variant Workbench: the data and the environment to compute on. One of the major benefits of the Kids First Variant Workbench is that Kids First has gone ahead and harmonized and processed all the data for you, so it's ready to use across all cohorts. And you don't have to spend the time to filter and do the pre-processing that you would otherwise have to do. We've made a data dictionary available, and I'll show it to you so you can see all the data that's there. but I do just want to call out some of the common tables that you're going to use in most, if not all, of your analysis. The occurrence table, which is there on the top left of the slide and has a little control thing, a little control tag, is really that table that links the variants to the participants. So you can only access this for studies that you have dbGaP approval. That's the key piece really that we can't display on the portal because it's not public information, but you can use it in the variant workbench. The variance table is more aligned with what you see on the portal. It has a list of variants and some of the common annotations you might use. There are many more tables, but patients and phenotypes allow you to find that row level information. Once you've figured out which participants have variant you're interested in, you can use additional tables to figure out the characteristics of those participants. And I'll show this shortly, but launching the variant workbench is as simple as launching a data studio session in Kavatica. The environment is custom built for this purpose. We've created a public project in Kavatica that has all the information you might need to get started and to read more about it. And there's a convenient link in the portal that'll direct you to that. Before the live demo, I want to bring it back to the scientific use case that we've been talking about. The demo will cover where to go, how to launch the variant workbench, all these things that you need to know. But we're highlighting that we now have this capability that we didn't used to have. The variant workbench is built in Jupyter, so there is a bit of coding, but I don't want the simplicity of the analysis to get lost in the code that I'm showing you. And so I just wanted to bring it back to this and say they're really simple steps that you go through when you use the variant workbench, which is starting the variant workbench, setting some filtering parameters, loading tables, joining, very basic commands, and then you can get your results all in one place in a very, I want to say easy, but very streamlined fashion. So I'm going to change over to Kavatica real quick. I think everyone's still seeing my screen. And what I want to do is show you, if you're not familiar with Kavatica, we have resources to kind of show you everything. But if you go to the public projects page, there's some analyses and things that are ready for you to look at. The first one that's going to be there is this Kids First variant workbench. There's a dashboard on the left with helpful information about getting started. And you can copy this and use it as your own project by clicking on this little eye and going to copy project. If you're using only open access information, you can actually go straight over to Data Studio and launch the variant workbench. But what we suspect is that most people are using this because they have controlled access. So to get that controlled access into your project, you can go, as David showed, go to the portal, click Launch in Kavatica, and it'll prompt you and ask you which project you want to load the data in, and you can select this project that you've just copied. I do want to scroll down and just point out a few things. Again, there's some information here, but the last bit is because we're a cloud compute platform, the analysis does cost something. However, Kids First and the NIH are very supportive of researchers and they want people to use this feature. So there are cloud credits available for you to help cover the cost of your analysis. And if you go to the bottom of the public project, you can get a link here. It's a fairly straightforward email to the cloud administrators, and then you can essentially do your analysis for free. I'm going to show a few things that you might find useful. So first of all, if you go over to Data Studio in the public project, there's going to be one Data Studio session that's preloaded with a handful of notebooks that are helpful for you. So I just want to show what those are briefly. We're running out of time, so I'm not going to go too much into them. But you have a handful of notebooks that you can look through. If you open up the session, you'll find them here and you can click on them. I think one that's very useful is this data dictionary. And you can see essentially all of the files that are made available to you in this environment. And you can actually also see the schema so you can understand what columns and what data is contained within those files. And if you scroll down, you can see there's Kids First files, there's open access annotation files, and that type of thing. I'm going to skip over to this notebook where we essentially recreated the analysis from Dr. Rao's paper. And I've hidden a lot of the code because, again, I want the message to come across that you can do this in this environment. But I'm going to highlight a few of the steps along the way. The first one, which is pretty much always, is to initialize this environment. So it essentially starts up a Spark cluster that you can then do that computational heavy activity on. Then you load the data. So in this note, or in this block, we're setting some filtering parameters. So here we wanted to look at the specific gene, AMOTL1. We set a minor allele frequency cutoff, a CAD cutoff, and then set some columns of interest. So we don't actually need to use every column of every table. We're only looking at certain information. This block of code loads tables into your environment. And this will be available for you so you can use as a reference. There's also, as I showed, other examples. So you're not coming up with this code from scratch. We really want this to be accessible to anyone. And really in this environment, there's only a handful of commands, and we've given examples of pretty much all of those. So it shouldn't be too onerous to kind of conceptualize what the analysis is and then copy the code for it. I've skipped over loading the other tables because it's essentially the same code, but you can see here we've loaded a handful of tables, including the occurrences table that connects those variants to participants. And then just as an example, we've aggregated phenotypes with participant ID, and you can take a peek at the top couple lines that show participant and then which phenotypes they have. I'm going to scroll down to this section that says replicating findings in the paper. And again, it's essentially taking some of those filter tables that we've had from before, joining them together, maybe doing some additional filtering. And what you can end up with is printing this table. And then if you so choose, you can reformat it. And the reason we reformat it is actually to look very similar to the table that was published in Dr. Rao's paper. So previously, whereas we couldn't do this in Cavatica, now you can do all of this in one environment. We then wanted to take this one step further and say, instead of looking in only 3 of the studies on Kids First, what if we looked across all the studies within Kids First? So we did this and looked specifically at phenotypes of those subjects that have that variants that meet the criteria. So I'm just going to scroll down to the end, and we've plotted at a very high level which phenotypes are present in participants across all Kids First studies that have variants of interest. So I'm actually going to stop it there. I'm going to go back to the slides quickly, and then we can wrap up. I do want to thank everyone that was involved in the webinar today. I also want to thank the teams that have been working really hard on the variant workbench. It's been a long time coming, and we were really excited to present it today. You can go on to Cavatica and Kids First today and start using it and playing around with it. And this last slide I have is really a call to action. We built these things for you, so we want you to go on, try them out, use them for your research. And if you need help, always reach out to us, and we're happy to chat.
Thank you,
so much, Jared and David.
Oh, and Adam. So I want to thank you all the speakers, and especially special thanks to Adam for his vision and his team at the Data Resource Center at the Children's Hospital of Philadelphia for making it a reality and bringing these amazing tools and resources to the Kids First program. So I have a question for David and Jared before we close, and we are over hour. So thank you for those of you that could stay with us a little bit past the hour. And also we will have it available, a recording of the program for the ones that could not stay. But Jared and David, for the people that will be interested in going deeper and continue, as I think we probably need a full hour for this demo next time. Do you offer like any support, office hours that you'd like to talk about before we end the program?
Thanks for that plug, Marcia. Yes, so once a month we offer Kids First DRC office hours where me and Jared and the portal team and the Velsera team are available to answer questions. So the Velsera team is actually available every week, but once a month on the third Thursday, we offer joint office hours which have a special Kids First focus. So it would be great for the next joint one is in June, the third Thursday in June. We'd love for folks who are looking to or maybe have had a chance to explore the workbench a little bit and want a deeper dive into that. I believe Jared and I will both be there, and we'll be able to go in much more depth, much more hands-on. We'll be able to answer questions about folks if they have it pulled up on their own screen. And so we would love to help people one-on-one, and we can provide information in our follow-ups for the information about the office hours, but the link is on our help center, the Kids First Support Help Center, and we'll follow up as part of our workshop today.
Thank you, David. And Adam, would you like to say anything?
Just gratitude for our speakers. Thanks so much, Dr. Rao, for joining today and for your work, and really for inspiring, as you saw, you know, really a lot of the dedication and effort of the team on behalf of creating new tools and resources for people like you. So thanks so much. And thanks again, Marcia, and to all the NIH staff and team for making this possible.
Thank you. And any questions before we close from the audience? I think people are leaving as we pass the hour. So again, thank you all and see you next time.
Look forward to it. Take care, everyone.
Thanks,
Bye-bye.
everyone.
Thank
you.
you.